Welcome children. We completed first two experiments and the third experiment is study of water samples. Okay, write the heading study of water samples. Then no need to write these introductory part. Then write objective, requirements, procedure, then observation, conclusion and precautions. Children, you have to draw this observation table and this diagram on the right side. That means on the blank part, you have to draw this observation table, then this diagram. Clear? Then experiment 3B. In next sheet, you can write experiment 3B. And this is to study the pH of various water samples. Write the objective, requirements, procedure, then observation and conclusion, precautions. This observation table on the right side. Clear? That means on the blank sheet. Then experiment 3C. Next page you have to write experiment 3C. Write the objective, requirements, procedure, observations till observation. Okay. So please leave one page for the drawing. No need to draw all these diagrams. Okay. After reopening, we have to select some of the diagram depending upon our experiment. Okay. So now no need to draw these diagrams. Clear? Experiment 3 is clear. Then experiment 4, experiment number 5, experiment number 6. These three topics deleted in the revised syllabus. Okay, so experiment 4, 5, 6 is not there in our new syllabus. So as experiment number 4, you have to write mitosis. Okay, so experiment number 4. This is experiment number 4. Prepare a temporary mound of onion root tip to study the mitosis. That is the heading. Okay. Then leave these landmarks. No need to write these landmarks. Then write the objective, requirements, procedure, observations, then interphase, prophase, metaphase, anaphase, telophase. You have to write all these notes and these diagrams on all these diagrams on the left side. Children, keep it in mind. In the first page, you have to draw one more diagram. It is not there in our lab manual. So in the note session, I will add this page. These diagrams, I will include it. Then all other diagrams are there in our lab manual. See, all the descriptions here and all the diagrams here. Clear? Mitosis clear. And after completing telophase, at the end you have to write the precautions. These part also I will give in the note session. So in this experiment, two extra parts are there. This diagram and this precautions. Okay, these two pages I will add in the photographs of these two pages I will add in the note session. Clear? Fourth experiment is clear. Next one, this is the fifth experiment. In our lab manual, this is the heading. Effect of temperature and pH on salivary amylase. Children, in the revised syllabus, temperature or pH, that is the name of the topic, effect of temperature or the pH. So any one of it is required. So we prefer effect of temperature. So no need to write pH. Okay. So in the heading you have to write to study the effect of temperature. No need to write three different pH. Okay. Temperature on the activity of salivary amylase on starch. Children, it is a major experiment. It is one of the major experiments. Okay. So, in our laboratory, we are taking three different temperatures. Normal temperature, very low temperature and high temperature. Okay. 2 degrees Celsius, 37 degrees Celsius and 70 degrees Celsius. But the procedure is not from our lab manual. We have to write the procedure as separate experiments. That means we have to write this experiment as three parts. That means the first experiment, fifth one, first experiment is normal temperature, that is 5A. Then it's a big one. Okay. Then 5B, action of salivary amylase at high temperature, that is 5B. Then this activity on cold temperature, that is 5C. Okay, so please don't write this experiment from our lab manual. Instead, I will add these photographs, okay, in our note session. Normal temperature, the activity in normal temperature, in high temperature, in cold temperature, we have to write. Then no need to write activity of salivary amylase on pH, because we are opting only different temperatures according to the revised syllabus. Okay, children. Then the last one is isolation of DNA. Isolation of DNA too we are writing from our record book. Isolation of DNA from plant material. I will add the photographs. Okay, isolation of DNA. Okay, so the experiment area is completed now. Then next class we can discuss the note session. That means in our question paper, the last session is sport at site. Okay, so different specimens we have to write, different disease causing organisms, and some Mendelian disorders too, that we can discuss in next class. So till isolation of DNA you have to complete now. Okay, children.